Alright, so today what we are going to do is we are going to, so yesterday the main theme, there were two. One, how changes in development enables evolution, and second we looked at cell lineage and fate mapping, right, the various techniques involved in it. So then I ended up saying that in the next class let us look at how development is controlled. So that is what we are going to learn today. So much of what we are going to go through today is again refreshing the history, but my firm belief is that helps in shaping our own thinking of development. Okay. So you might think, oh, these are all history, it's over, and what's the point of knowing, we know now the truth. That is an argument, a valid argument, but I still feel it is worth taking a few minutes to look at the history, how the thought shaped up in a given field. So therefore here, like epigenesis and pre-formation, we are going to get into another controversy that was laid to rest, believe it or not, only a few years ago. Okay. In about 20 years ago only the ultimate proof came for that. So we will look at that. So most of you already know all cells have same set of chromosomes, and that is from where this concept called genomic equivalence comes. What it means is you take any cell of the adult body, they have the same set of chromosomes. There are exceptions. Our RBCs do not even have a nucleus, and if you were willing to think a little bit more, some of you may have already done an immunology course, right. So have you learned VDJ recombination? But at least you will know this, in response to a foreign material, our B cells are capable of generating an antibody that is customized for a particular pathogen, right, particular shape actually, it is not even pathogen. So how is that being made possible, right. So their genome again undergoes rearrangement, that is what is VDJ recombination. So each B cell therefore has a genome that is different from another B cell, leave alone another somatic cell. So there are variations, but by and large most of the somatic cells in any organism has the same set of chromosomes, and that is what we call as genomic equivalence. So now the question is what controls development? Is it the cytoplasm of the oocyte? Oocyte comes with a huge cytoplasm, it is much bigger than most of the other cells. So does it come with all the required thing for an organism's development? So that was one school of thought. Another one was chromosomes, it is the chromosomes that determine, not the cytoplasm. So then people pursued both of this to find evidences, that is how science progresses. So we will see those. So one of the really famous embryologists, he has contributed really substantially to the field of development as well as genetics, and his name is Theodore Boveri. So one of his evidences is what we are going to look at, that is, so he looked at cytoplasm. Okay, we are going to skip some of the genetics here, that is, how did people find Mendel's genes are located on chromosomes, okay, that we are ignoring. So in genetics class we can learn that. So here we are assuming that we already know that genes are on chromosomes. Very briefly, one of the things was the behavior of chromosomes when cytologists looked and observed the chromosomes were following Mendel's laws. Okay, so that is very briefly one sentence description of that evidence, and there are more, some of them we will see here too. So what he did is, he took this sea urchin, have you seen them? So even a 10-minute walk down the Elliott's beach, you will pick them on the sand. So I have done it multiple times. I have done in Elliott's beach as well as I have done in Thiruvaluvar beach, and I have done multiple times in Mahabalipuram. Okay. So next time when you go to beach, just walk along combing the seashore you will find these. So, they are there, but many of the mollusks, crustaceans, you can find them on the beach, live organism. So, you can observe them. So, that is interesting, and you can see huge turtles and nesting, that also you can see. So, he took sea urchin, and this is external fertilization. So, a lot of people used sea urchin for studying fertilization. So, that is another topic, okay, very in the later part of this course we will learn that. So, he fertilized with a large excess of sperm such that he got some of the zygotes with more than one sperm, like as you see here, okay, 2 sperm, and then he allowed to embryogenesis to progress, and it sets up 4 poles like 2 spindles, and therefore, it ends up making all kinds of weird spindles and end up dividing into 4 cells. At a point it should have divided into 2 cells, and each one having varying numbers of chromosomes, and he isolated blastomeres and showed when chromosomal abnormalities were there, they just did not progress further in cell division and died. So, here you are seeing that an embryo disintegrating cells and death of embryo. So, this is one of the evidences indicating that normal embryogenesis requires normal number of chromosomes. So, therefore, chromosomes matter. Okay. And 2 other scientists, Nettie Stevens, she was a postdoc of T.H. Morgan. He is a very famous geneticist, but originally he supported cytoplasmic control of development. Yes. So, the embryologists were of 2 schools at that time. So, one of T.H. Morgan's postdoc, Nettie Stevens, she strongly felt it is the chromosomes, and she went and worked with this guy Edmund Wilson, who also believed in it, and they showed in many organisms sex determination is by the chromosomes, XY or X0 in many organisms are male and XX was female. So, there is another support, and ironically you see the way science progresses. T.H. Morgan tested this more rigorously and nailed down the Drosophila eye color gene is inherited on an X chromosome basis. It is a sex linked inheritance, and that eventually proved genes are on chromosomes. So, that is how science progresses. People do not get stuck on an idea forever. When evidence is surfaced, they change their opinion. Okay. So, this is some interesting history to know. So, I am assuming some of you are going to be, some of you are budding scientists, and therefore, the way thought process shaped up in the past scientists will be useful. So, that is one part. So, these are all evidences that are being stacked to show chromosomes matter, and you are obviously now having a question. The question is, if all cells have the same chromosomes and chromosomes direct development, then how different kinds of cells come? How come the same set of chromosomes make my heart and make my lungs as well, right? How is that being possible? So, that is a big question. So, August Weismann proposed a theory called germplasm theory. Okay. So, this is one of the really serious milestones in biology. So, many biologists consider this germplasm theory, its importance as next only to Darwin's theory of evolution. So, what is his theory? He is the first one to distinguish somatic cells from germ cells. So, he proposed that the germ cells maintain all the chromosomes, the genetic material in its entirety, and that is passed on from generation to generation, continuity of germline. Okay. So, whereas the somatic cells that come out of these germ cells in every generation, that is germ cells fuse like the gametes fuse, generate the zygote, and from the zygote you get germ cells as well as the rest of the body, and the rest of the body, for example muscles, receive only the instructions needed to make muscle. and blood cells receive only the instructions to make blood cells. Neurons receive the information to make neurons. So, this is what is germplasm theory is, and this is how he thought different cells become different kinds of cells. So, Bovary's further experiment. So, this was a major milestone, okay. Because this the reason this is major milestone is this helps us to distinguish Lamarck's view of evolution from Darwin's view of evolution, okay. So, use-disuse theory would demand the information in the muscle goes to the next generation. So, that is the only way learned characters can be acquired, and germplasm theory explains why that will not happen, because information from muscle does not go to the next generation. So, only the germ cell goes. So, therefore, random selection from changes that happen in the germ cell chromosomes is what gets selected in every generation, okay. So, that is how this germplasm theory helped in understanding Darwin's theory of evolution, okay. The main component of Darwin's theory of evolution is not that he first told evolution happens. The main contribution is finding a mechanism for evolution. So, the mechanism is natural selection, and for that this was very useful to explain why acquired characters are not going to be passed on directly to the right next generation. So, Boveri did additional experiments that sort of supported germplasm theory. That is primarily because of the organism he chose. So, C. elegans was not the first nematode that was picked up for studies. So, in 1901 Boveri was using Ascaris, but you need to remember Ascaris is a parasitic nematode. So, finally Boveri died of infection by Ascaris years later, okay. He had a long life, but died of the model killed by the model organism. So, the reason he chose this is it has only 2 chromosomes. So, again you see this, for addressing specific biological questions you need to have knowledge of organisms, okay. So, then only you will know which organism is suitable to address a specific question, without which you would just not be able to answer. So, that is why it is more important to learn botany and zoology before actually you learn molecular biology and biochemistry, okay. So, that is why we will not find C. elegans, we will not find Drosophila, we will not find Arabidopsis, because we do not learn organisms. So, he knew this has only 2 chromosomes, and therefore, it will be easy to observe what happens to these 2 chromosomes as cells divide. So, Auguste Weismann's theory proposes that the somatic cells will have different contents, okay. Is this happening to these 2 chromosomes? And it did happen, okay. So, as only 2 chromosomes, easy to follow, and here are the cell divisions. Some of you who are learning C. elegans would see that the names he uses is what we use in C. elegans also, like EMS, endoderm muscles come from that, P1, P2, P3, P4, the germline lineage. So, that is how he named them. So, here if you look at it, in the first one, this bottom one is going to be the future germline, okay. So, you have the chromosomes here, and go to the next one. When this top cell is setting up the next spindle for the next division, you see the chromosomes are sort of breaking in. So, this is what is happening, while here it remains intact, and you go through this over division. So, this is the one that is going to be germ cell, this is the one that is going to be germ cell, this is the one that is going to be germ cell, and you look at the adjacent cells, the chromosomes break. So, it's the compared to this is the most recent one that came out of this lineage. Here it is not fragmented so much, but in earlier somatic blastomeres you see it fragmented very strongly. So, this process is called chromosome diminution, okay. And you go further in a embryo where the cleavage is over, the 2 primordial germ cells retain the chromosomes intact, while in others it has broken into smaller pieces. So, this is a strong support for germplasm theory, that is okay. So, that is Ascaris, but is this happening in every organism, right? But this is not what happens in other organisms. So, that means there must be other explanations in other organisms to support germplasm theory, or germplasm theory may be wrong. So, therefore, the objection for nuclear control of development persisted. So, people still thought that nucleus may not be everything, meaning chromosomes may not be everything, cytoplasm probably still is the key. So, the only way to address this is to really take nucleus of a fully differentiated somatic cell and show that it can direct an egg into an fully developed embryo. So, that is what was required. Really what you needed is somatic nuclear transfer. So, that required some technical advancement, like you needed to find a way to take the nucleus out of an egg and at the same time activate the egg. So cytoplasm gets activated when sperm enters. So, you needed to do that. Then you introduce a somatic nucleus, and that had to be figured out, and people did that eventually, like Keith Porter developed a technique for doing this. So, he found that by poking in and trying to just move the nucleus out, not only removed the nucleus, that ended up activating the cytoplasm of the egg as well. So, this is called Potrust technique. Okay. So, he found a way to remove the nucleus from the egg, and making further improvements on that, Briggs and King, they were able to show that transplanted nucleus from other cells can actually make a frog egg develop normally. Okay. But the only difference is that they were able to go up to a certain stage, like up to blastula stage, but not up to the tadpole stage, and that weight needed some more optimization, and they had to work on a different set of frog. And so, here is an example of how this is done. So, this is actually a movie, but I am sure the movie is not going to play. So, essentially, so you have a skin cell, you have an egg cell, then you take the nucleus out, and then you introduce the nucleus from the skin cell and then allow it to develop. So, the way you do that is, so this is an egg. So, here you have a capillary where you apply gentle pressure, therefore, it is held in position, it does not move away. Okay. So, this technique will vary from organism to organism. This holds true for most of the mammals and even invertebrates. So, here is suction applied, so it is held in place. So, then this is the needle that you are going to poke in, and in that you are going to gently take the nucleus out, and the other nucleus that you get from skin cell, essentially what you are doing is you are putting the entire cell into this, and then its nucleus is going to be it is a diploid nucleus. So, you do not have sperm in this, and you watch what happens. So, there were two issues with Briggs and King experiment. One is that it did not go beyond blastula stage, and the other one is they used from another embryonic cell. Okay, it is still an undifferentiated cell, it is not equivalent of a fully developed somatic organ. So, John Gurdon, who got Nobel Prize just few years ago, less than 10 years ago, I forgot, but very recently, okay, got Nobel Prize for that, but the work was done in 1962. So, he got Nobel Prize along with the Yamanaka group for doing somatic cloning in mouse. Okay. So, but they did it very recently in mouse, but he had done in 1960s, and he showed that you can take intestinal cell nuclei and introduce by using Briggs and King's technique, and they can actually develop all the way up to tadpoles. And using different genetic makeup, like for example, here you have a dark colored frog, and you take the nuclei from this light colored one, and using its egg, then you show that all the progeny are like the nucleus donor, okay, showing that two things: one, nucleus directs development, and second, evidence for genomic equivalence, right. He is taking intestinal cells and they are having it, Of course, people have done with other somatic cells as well now, variety of somatic cells have been used. And this is one more evidence. So, this I am sure some of you were born by the time this was published, it is that recent. So, Wilmot in Scotland, so he and his group, they were able to do very similar experiment in sheep. Like this is a oocyte donor from which you are taking the egg, then you take it out the nucleus and the spindle, and then you take the udder cells from the nucleus donor, and then you transfer into the enucleated oocyte and then allow it to develop. Yeah. So, allow it to like you activate it by electric shock, so that allows the membrane fusion. Then you culture them in embryo up to this stage, blastocyst, and then you implant in the surrogate mother, and then the progeny is genetically identical to the nucleus donor, not the one that provided the oocyte. Okay. So, this clearly nails down that there is genomic equivalence as well as nucleus drives the development. So, this is the dolly and its own baby here. Okay. But this is not to say that DNA alone is responsible for development. Okay. So, there are variations to this largely true concept. So, those variations are subtle variations, but still important. One of them is illustrated here. Okay. So, this kitten is somatic clone from this cat, and in these cats the coat color has random variations, and that is because of random inactivation of one of the X chromosomes. But to get to that, there is a concept called dosage compensation, right. So, all the girls here have 2 X chromosomes and boys have only 1 X chromosome. So, are we going to have double the amount of X chromosome output, or are we going to have half of the output in boys, right. But at the end it is the same output, that is because the 1 extra X chromosome is inactivated. So, there are multiple mechanisms to handle dosage compensation, but we will not get into all of them. So, we will just consider the organisms where 1 X chromosome is inactivated. So, which X chromosome is inactivated and at what stage in development? So, that varies from organism to organism, and in this cat it is random inactivation in every one of the somatic cells. So, depending on in which part of the skin which X chromosome is inactivated, you get different color patterns, and due to that this kitten does not look identical. Okay. So, that is one, and so these are epigenetic modification. Okay. The DNA still remains the same, and DNA is largely responsible, and there are other situations like environment can still have influence. So, DNA is not the sole determinant, but for the original question the answer is yes, it is the set of chromosomes that determine development. So, this is actually how we learned chromosomes direct development, and the genetic content is of most cells are equivalent. I am saying most because there are variations like our RBCs, immune cells. So, if that is the case, then same set of chromosomes, how they direct development, each cell has to become different kind of cell, how does that happen? So, therefore, the postulate here is development must proceed by temporarily inactivating parts of chromosomes, because the chromosomes still exist, there is no chromosome diminution as we saw in Ascaris. So, that means part of the chromosome is expressed in one type of cell and another part is expressed in another kind of cell, and so on. And that is what brings us to a concept called differential gene expression. So, most of the modern developmental biologists deal with this idea of differential gene expression. So, that is what people focus on, much of the research is about that. So, genes control development, and if they are about to, what is the answer? It is the differential gene expression. Okay. So, that is the end of this discussion. So, we will move on to our next thing. So, we will look at how the differential gene expression actually happens, since we have few more minutes. So, gene expression actually can be controlled at different levels. I am sure many of you may be already familiar from your molecular biology class, but just to give continuity I will quickly go through them. So, you can have differences in gene transcription. For example, globin gene is not transcribed in pancreatic cells which produce insulin and other cells do not produce them. Okay. So, that is at transcription level. So, a gene may be transcribed in one type of cell, but not in another type. So, that is differential gene transcription. And second, similar difference may exist at the level of nuclear RNA processing, for example, splicing and export out of the nucleus. So, that might vary. In a given tissue, a particular gene's mRNA may not be exported out, while the same mRNA is exported into the cytoplasm in another type of cell. So, that is selective nuclear RNA processing. And then you have selective messenger RNA translation. For example, if you take two tissues like the germ cells or neurons, there the response required is really quick. Like if you take neurons for example, the response required is really quick. So, you can't be activating the expression of a new gene all the way starting from transcription onwards, you may not have the time. So, in such situations you have mRNA is already made, but not translated till it is needed. Okay. Similarly, if you take reproductive the biological process of reproduction, early embryonic development will not have during the rapid cleavage will not have the ability to do everything by starting from transcription onwards. Lot of proteins required during early embryogenesis, their mRNAs are already transcribed in the mother's germline and brought in via the oocyte cytoplasm, where the mRNA are kept translationally quiescent and their translation is sequentially activated as and when they are required. Okay. So, these are two very good examples where translation control plays a major role. So, that is how you have selective messenger RNA translation. So, that is another step of gene expression where you can have control to bring about differential gene expression. And another important step. So, these are the four really key steps, but there are other sub steps in each one of these where you can have gene expression control. So, do not think gene expression control means transcriptional control. That is a common misconception among many students who have not studied developmental biology. Okay. But after this course you are not going to have that feeling. And you can have at the level of protein modification, right. So, you have seen two kinds of protein modification if you have attended the biochemistry class. One is zymogen activation, like protein is made pre-pro protein and then it is cleaved to generate the final product, like some of the hormones and blood clotting factors and so on. And then you have other proteins where post translational modifications such as phosphorylation activate or inactivate proteins. So, these are the levels at which you can actually make differences among the cell types in terms of what genes are expressed and what genes are not expressed. So, we are going to look at these regulations, and before we go into this, we are going to have a good idea of chromosome structure and a good idea of eukaryotic gene structure. Okay, that is what we are going to do in the next few slides. So, I am sure you are familiar, but I just want to quickly go through this. So, this is crystal structure of the same here. Okay, so, it is the chromosome with all the proteins are histones here, right. So, you have the different histones, the 4 different ones differently colored here, and the DNA is wrapped around it. And pay attention to these tails. So, these are histone tails, okay, coming out. So, they are important for our discussion. And you have one histone. So, this orange rod like structure. So, that H1 that plays a key role in really compacting this structure into a long spring like structure. Okay. I am particularly and intentionally avoiding the word solenoid, because some of you will have to go and look at the dictionary to find what solenoid mean. It means a spring, okay, a coil. So, this coiling is possible because of the H1 that binds here, that can actually pull them together and make them become a coil like this. So, this is how our chromosome exists, tightly coiled condensed structure. So, it is a a super coiling, because here itself you have coiling and then you have additional layer of coiling, and each individual one is nucleosome. So, they contain 8 molecules of histones, H2A, H2B, 4 of them, then H3 2, H4 2. So, that is the structure here, okay. And pay attention to these tails. So, this is how a chromosome exists. Now, let us pay attention to these tails as So, it so happens some parts of the chromosome is tightly condensed and we call them heterochromatin, and there most of these tails on H3 and H4 are methylated, and the methylation normally promotes this coiling, and these are usually transcriptionally silent. The transcription factors and RNA polymerase do not have ready access to this, okay, unless otherwise a special kind of transcription factor called pioneer transcription factors that we will learn later when they come and open it up. And in uncondensed nucleosomes, where you have this one single structure is a nucleosome, okay. In uncondensed nucleosomes, the histones lack the methylation marks, not all of the methylation marks, but majority of these, and they are usually acetylated, okay. So, acetylation in H2, H3, H4, so that marks active chromatin. So, here you are seeing a control of gene expression at the anatomy of the chromosome, okay, and it is largely regulated by modifications to these tails, and that is why I was telling pay attention to the tail at the crystal structure level itself. So, this gives you an idea of how they are available readily, or rather readily accessible for enzymes that could add or remove methyl group or acetyl group. So, now a closer look at one of the nucleosomes, particularly focusing on H3's tail. So, here if you look at, you have, so these red ones are the methylation marks. So, some of the red methylation marks combined with acetylation in general activate transcription. So, that is the point I want to make here. It is not that methylation means inactivation regardless of where the methylation is. So, here these numbers 79, 38, 27, 9, 4 etc. Refer to lysine residues in H3 starting from its N terminus to C terminus. So, depending on which one of these lysines are methylated and whether the overall chromatin is acetylated or not determines whether it is going to be active or not. So, these are shown here. For example, when you have H3K4, okay, H3 lysine 4 is methylated, then this factor is going to bind and that leads to transcriptional activation, okay. And here H3K9 methylation silent heterochromatin, okay. So, this is how modifications to histones can have an impact on gene expression. So, here the control is at the transcriptional level, okay. So, the chromosome anatomy itself affecting transcription. So, we have another layer that affects transcription. For example, and also to avoid confusion, you need to remember, in addition to methylation, which is a post translational modification of the histone, which is a protein, you have methylation of nitrogenous bases on DNA as well, for example cytosine methylation. So, do not get confused between the two methylations. So, in a chromosome DNA can be methylated, which is not our current discussion, and you can have proteins that are methylated as well. So, here we are talking about protein methylation, and the protein is histone. Clear? Okay. So, having looked at the chromosome as a whole, now we will refresh our memory about the structure of a eukaryotic gene. So, I do not understand what is the problem, but most students, after going through molecular biology course, even after a couple of years later, when you ask them to draw the structure of a eukaryotic chromosome, they have gene, they have problems. And people do not understand, for example, where is a promoter with respect to start codon and what is 5' untranslated region. Okay. Is 3' untranslated region present in the chromosome or not, and is the poly A tail present in the chromosome, or where does it come from? So, like that people have confusions. To avoid that, we are going to refresh our memory of the structure of a eukaryotic gene. So, this horizontal bar represents the DNA sequence, okay, of a part of eukaryotic chromosome. So, here first we will focus on what is easy for us to understand, that is the coding sequence. So, the coding sequence is divided into exons, meaning there are intervening sequences that are not going to be part of the mature mRNA. So, they are intron 1, intron 2 here. So, you need to get rid of this in the final mature mRNA and have the sequences corresponding to this exon 1 in the DNA, exon 2 right after that, and exon 3 right after that. So, then only starting from the start codon, which is ATG coding for methionine, you go all the way to stop codon. So, when you look at the gene structure, these coding sequences are divided into exons with intervening introns. Is this clear? So, this we understood now, starting from ATG to stop codon. Okay, stop codon somewhere here. Okay, so this 146, let us say. So, then when you look at the 3' end, you have further sequences, and they are called the 3' untranslated region, meaning these are present in the mRNA, mature mRNA, but they do not code for amino acids. These are sequences beyond stop codon. So, the primary point I want to emphasize is the mRNA has sequences beyond the stop codon, and that is called 3' untranslated region, because in terms of the directionality of the mRNA, it is 5' to 3', so it is 3' untranslated region. So, obviously that is coming from the chromosome, it is transcribed from the chromosome and it is retained in the mRNA, therefore, it is an exon. 3' UTR corresponds to an exon, just like the amino acid coding sequences. And in that RNA sequence, that 3' UTR, you have sequences like polyadenylation signaling sequence, which signals two things: one, cleavage of the RNA from the primary transcript, and then promoting polyadenylation, like poly A tails are added. So, some amount of poly A tail is added in the nucleus, and further extension of that happens in the cytoplasm. Okay. So, poly A tail is not present in the chromosome. It is a separate enzyme that adds multiple A's continuously one after the other. It does not require a template, because you are just constantly adding A to an existing RNA. So, that is how that is made. So, the summary is that after stop codon you do have RNA sequence, and that is called 3' untranslated region, and that contains a polyadenylation sequence that helps in cleavage of the primary transcript at the 3' end, addition of a poly A tail. And let us go to the 5' now. So, in the 5 prime similarly, before the ATG you have sequence, and that is called 5 prime untranslated region, and that is that usually starts with the base called transcription start site, and that is modified like the 3 prime modified with poly A tail. This gets a cap, 5 prime cap, okay. It is a trimethyl G added to it. So, that is the RNA sequence, but here in the DNA sequence upstream of the ATG you are going to have some RNA sequence, okay. That is also part of the exon, that is also in the genome, while the trimethyl G is not part of the genome. So, then upstream of that you have promoter, okay. So, promoter is where RNA polymerase is going to bind and start transcribing, and the first base that is transcribed is the transcription start site or the initiation site, that is not ATG. ATG is going to come further downstream at the end of 5 prime untranslated region, and the promoter region can have multiple parts in it, which we will discuss in detail as we go. Tata box is one that is present in all promoters. So, these are like core promoters, promoter sequence which is required for transcription of any gene, and there are special sequences that help in differential gene expression. So, that we will talk about it when we go further.